Okay, so we will start. Welcome to the introduction to modeling for synthetic biology, the last webinar of the measurement committee. I'm Alejandro Vignoni, and I will try to explain you a little bit. Sorry. First, what is a model, and where is modeling and the design, build, and test cycle of synthetic biology, and finally, of that part, why it is important to model. Then I will deal a bit with what we will want to model. I will give you some examples of models in synthetic biology, and then we will talk about a bit of reporters and calibration, which we deal with that in the measurement committee. Okay, so the survey, if we didn't fill it, please. I will take a look at the results. Okay, so 70% of the list of the people attending is iGEM students from this year, so very good. And the backgrounds that I see here, there is a lot of biology, biochemistry, also some engineering and computer science. So we have a pretty good background. Okay, all the results will be used later to decide what we will do in the summer with the measurement committee, but I will tell you later. Okay, so you told me in the survey who you are. I will tell you who am I a little bit short. I'm Alejandro Vignoni. I studied electronic engineering in Argentina. I finished in 2009. Then I went to do my PhD in Spain, in Valencia, in control engineering. And I found iGEM in 2010 as a student for the first time. I went as a student. And then I kept my relationship with iGEM as a student, then as advisor. In 2018, I was instructor of the Valencia UPV team, where we won the grand prize. We were very proud. And from that also, I started to be a judge and member of the measurement committee since the last year. And that's what brings me here. Okay, so two important things that we want you to get from this webinar. First, why it's important to model and design before the implementation and before going to the lab. This is particularly important and interesting this year because many labs in the world we know are not able to go to the lab yet because of the COVID pandemic. So it's good that we take advantage of this situation and learn how to model and design before going to the lab. And the second thing is how to start modeling for synthetic biology in the second place. Okay, so first, so what is a model? I will give you some examples. Some are a little bit a joke to lighten the mood. Models. These are models, for example, fashion models. Also, different kinds of models. Architectural models that architects build before going to the construction to see how everything will go beforehand. Another model that is very famous is the zebrafish. It's used by biologists to study other organisms. Is what is called a model organism. So to sum up, what is a model? A model is something that represents another system that we want to study, and the ones that are relevant, the models that are relevant for what we want to do, I will show here. So there are different types of models. First, the schematic models, we are very used to these ones, are little sketches that tells us interactions between elements of the system. This is an example of a synthetic circuit. Then, from those schematics, we can go on and try to write down the biochemical reactions underlying that system, that will tell us a bit more information about the kinetics of those species, how they change. And from these reactions, we can go and then build us a mathematical model, basically a set of ordinary differential equations done be scared, that using the law of mass action kinetics, we can convert from the biochemical reactions into these sets of equations that will tell us basically how are changing in time the different amounts or concentrations of the species in the system. Okay? So how do we start? In general, I've seen this picture many times. We go to the lab, we build our circuit, we get many experiments. For example, plate reader experiments, also flow cytometry experiments that gives us information about fluorescence and the circuits in the cells, microscope pictures, etc. And what is general the case? What happens? People say, okay, let us make a mathematical model to fit all these experimental data and to see what happens. But let me tell you that this is not the way we want to do. I will take this out because this is not the way we want to go. Why? Because we want to work in synthetic biology using an approach which is based on all the engineering disciplines, which is the design, build, and test cycle. If you were yesterday in the webinar by the measurement committee, Ed was talking about how if a civil engineer was a molecular biologist to go and then build a bridge, the bridge doesn't work, then you go and you build it again and again and again until it works. That is not this design, build, and test cycle. What we want to do is to first design, to have the idea of what we want to do and to model the system that we want to build. Then we go to the lab and build it. Yesterday we had the seminar from the measurement committee also on assembly and cloning a little bit. And then once you have your circuit built, you can go and after having some calibrations, you can measure and test your system to see how it works. And then, as this is a cycle, you can go back, and if it didn't work like you were expecting, you can redesign and go again. So, but why is important to model? When we put modeling in this cycle, what is allowing us is to prove that an experimental design is feasible. Also, it can guide us in the selection of parts and assembly. We will see a bit of this. We will be able to predict the expected output of our circuit before going to the lab. That one is important also. And when we cycle back, it will provide us maybe with a better design that we didn't think in the first place. We will be able to redesign our original design. Okay. This one has a lot of text, but I will just go by few of them. Sorry, but it's to be them written there, so we can go back if we want to read them. But basically, I will pick the third one, which I think is important. A mathematical model will allow you to perform experiments in conditions that would be dangerous or of high economic cost in the real system. And this basically is a kind of troubleshooting before the construction and the assembly. You can try different things in the model and see what happens. And then you can go back to the lab and select the one that did work for the construction. In the old times when modeling was not around in these areas, it was the normal workflow, was to build, test, doesn't work, you go back, you build again, and on until you find the one that works. With models, you can do this troubleshooting in the model, try to build the different alternatives in the design, and once you find the one that you like, you can go and build that one, saving time and money. Okay, so I leave them here for you to read them later when this is finished. So now, we gave the introduction. Now, what are we going to model? So the first things that has to come back to mind, many of you know it, but I will remember it. So basically what we will try to model is the central dogma of molecular biology, which is what? Information is stored in DNA. Jake was talking about this this morning, about the information processing. And then RNA polymerase converts that into mRNA, and the ribosomes translate that into proteins, which are the elements that take action in the cell and responsible of many of the functions of a living organism. So we go from the information to the action, from DNA to protein with this process. So, but putting that more into context of iGEM and synthetic biology, in order to that process to happen, we need to have these 4 basic elements in a device to be able to the polymerase to find DNA, do the transcription, and then the ribosome find the RNA and do the translation. We need these 4 basic elements. We were also talking about this in the previous seminar yesterday, Alexis and Ian. The 4 elements that we need: promoter, RBS, coding sequence, and terminator. The first 2, you can think of them as control elements. The promoter will control the relationship with the RNA polymerase, and how translation happens, transcription. And then the RBS controls the initiation rate and the binding with the ribosome and affects more the translation. Then, of course, the more important element is the coding sequence. It will define what protein we want to produce, and the terminator to finish the transcription. And then, as Jake was talking this morning, we will like to go up in this ladder and be able to build systems with devices and transcriptional units to be able to have a function. But for now, we will concentrate in these 2 elements of the bottom, which are the ones that are more relevant to us now. So how can, probably most of you know, but just a reminder, how can gene expression be? It can be an unregulated process, which is what is called a constitutive expression. The gene is always on. This process that we were talking before is always happening. RNA polymerase is transcribing, ribosomes are translating, and we produce protein. And the second option is that we can have, we can have a regulated transcription and translation process, and that will lead us to the option of having the gene on and off, and to decide when we want gene expression to happen. And so let's go a little bit more deep on that. The regulated version that we have, when we want to do that, will involve some proteins, which are the transcription factors, which will bind to the promoter and inhibit or activate the transcriptions of this transcriptional unit, depending on the way they are. So, for example, so basically that's it. We have the 2 kinds of gene expression, the constitutive and the control one, and the control one will depend on the transcription factor, which in general is a protein or a collection of proteins that will tell us when this process wants to start or to stop. So let's go to understand a little bit more deeper what's happening here. We want to model, we will start with the easy one. We want to model this process that we were talking before. We have a gene here, which has a rate of transcription, which depends on many things, the amount of polymerase, the binding affinity of the polymerase to the promoter, and many other things. And with that rate, we will be producing mRNA. You don't see the pointer, don't you? So I will go to this one. Yeah, mRNA. And then ribosomes, as we explained before, will produce with a different rate, K2, will produce proteins. And of course, all the species, RNA and protein, will have degradation because they are not here forever. They will have degradation rate. So with all this information, using the laws of mass action kinetics, we can write the differential equations that model this behavior. So there is a lot of information in this slide, but let's go step by step. The first one, here we are having the two equations that describe the dynamic, the changes of RNA, which is the M for mRNA, and for the protein, the P. And this, what is written here, is the notation for the time derivative. This means the changes of the mRNA related to time, and this one is the change of protein in time. And what is telling us this equation is that the amount of RNA will be depending on this number, K1, which comes from here, is the transcription rate. And it will disappear, that's why the minus, at a rate which is proportional to this D1 value, which is the one that represents the degradation rate of the mRNA. Okay, so positive terms in these equations means that the amount of this guy mRNA in this case will go up, will increase, and negative terms will be responsible for the amount of mRNA going down. And then we can do the same with the protein. The amount of protein will change in this way, proportional to the constant K2, which is the translation rate, and proportional to the amount of mRNA M, which comes from the previous equation. This is the term that tells us how the protein amount will increase in time. And then the negative terms tell us how it will go down, how it will decrease, which will be proportional to D2 and the protein itself, which is the degradation rate. Okay. So what about those numbers? Where do we get those numbers? This is the first problem that we find when we try to go on with this. I will copy here in the chat the link to BioNumbers. It's a database collected by a group in Harvard, which is taking from many papers different values of concentrations and constants that are around, so we can use it for our models. For example, we can know that the half-life of a normal mRNA is around between 5 and 10 minutes. Also, that the half-life of GFP can be also around of 90 minutes. So we can try to go there and find values for the parameters we need. Okay. So we have here again our equations that represent the changes in mRNA and protein in the constitutive gene expression case. And here what I put is a plot of, we will talk later about this, the unit that we have on this side. It's amount of protein, or is proportional to the amount of protein in molecules of equivalent fluorescein per particle. We will go back to that. But also the same amount, the unit that I have here in the line is number of proteins. So what I'm showing here is 3 different cases of this model with different values of K2, which is the responsible of the translation rate, and that in the lab will be represented by different RBSs. So basically these are 3 transcriptional units: constitutive expression with a fixed promoter and GFP, and changing the RBS, we can get different levels of expression, and in the model that is represented in changes in the value of K2 that give you different values of the number of proteins here. But so this is just an introduction. We will come back to this later. Now, the previous things, the constitutive expression is a little bit boring because it's always on, as you saw in the graph. It's changing a little bit from the initial condition, but it's basically boring because you can't change anything. Let's go to a more fun way of regulation of gene expression, which is activators and repressors, as we said before. So basically, the equations look very similar to the ones before. The protein equation is the same. Protein changes depending on the translation rate and the amount of mRNAs, and it goes down depending on the degradation rate of the protein. But we have a difference in the equation of the mRNA. This part is the same. The degradation depends on the degradation rate, and the production of the mRNA depends on this equation, which is known as a Hill function, that we will see how it works a bit later. And if you see here, it depends on A. A is another variable in the system, which is the amount of activator that we have. Later on, we will be able to model also this A, depending on what's going on. But for here can be something that we can put from outside, and we will be able to see how this changes the level of mRNA. I see what is K again? Which K? There are many Ks here. Itmar is asking. K to the N. This one I will explain in the next slide. This one is the dissociation rate of the activator. We will see that in a moment. So exactly, to explain that, let me show you a little bit a small app. Do you all see this? So if here we have a different way of writing the equation, is the normalized Hill function. B0, this beta 0 is what is called the basal expression. Is if all this is 0, is what will be expressed without any amount of activator. A is the amount of activator. N is what is called the Hill coefficient, and KD is the half concentration constant. It's not the dissociation constant. And so if we change here, for example, we can change. Okay, we can change the value of the. Is it changing? No. Yeah, here is changing. So with this wheel, I can change the value of the half concentration constant, and we can see how that affects the curve. Let me explain a little bit the curve, because N is the Hill coefficient, is the exponent here, and is what represents the amount of transcription factors, of the cooperativity of the transcription factor that you need together to be active and bind to the promoter. So I see that this is bringing up a lot of questions, and we are thinking of making a more in-depth webinar in summer. And one of the things that I'm thinking on doing is to do all the equations that we need to get this equation, to understand exactly what are all these elements and where is this coming from. But for now, we will only explain how it works. KD is the half concentration constant. If you look, so it's related to the dissociation constant that Dogu says, but if you write the Hill function in this way, it's called the half concentration constant. And if you look at the plot here, for example, I will put to the value 400. We put 400 and we find 400, we will put basal expression to 0. So this is the ideal, sorry. Yeah, there we go. This is the ideal, for call it in a way, Hill function, when there is zero molecules of the activator, there is no expression. There is no value in the Hill function. When we increase the number of molecules of the activator, the number going out from the Hill function goes up, and it goes to 50%, which is here, at the value of KD, which is 400. Okay, so if you look, 0.5 is here, and this is 400. Okay, So what happens if we increase the number n, the Hill coefficient? To the Hill coefficient in nature, it goes, this is not working. Yeah, sorry, I have to take this one out. So if we increase the Hill coefficient, do you see that the slope here in the middle is getting more steep? This means that you need more molecules together. For example, a Hill coefficient of 4 means that we need 4 molecules of the activator together to bind to the promoter and activate the expression. So in nature, Hill coefficient goes from 1 when there is no cooperativity, from 2 to 4, depending on the biology behind. For example, the lac repressor is modeled with the Hill coefficient of 4. All that numbers you can find in the bio numbers that we showed before. Okay, so I will provide this the code of this app later. We will see how we in the measurement. Yeah, exactly like Alexis says. We will talk about later how we can give you this this information. Let's make it interactive. Victoria says if there is a free version of MATLAB. I think we have to check for this year, but I think MATLAB will provide licenses for iGEM for this year, but we have to check with the HQ, with headquarters. But I think that was the comment that I got from Tracy. But we can check and let you know later. Okay, so let's go on. So what happens if instead of an activator we have a repressor? This is the same, but instead of the transcription factor that comes here in the repressor, instead of activating, it represses. So if you look at the equations, they look very similar to the ones before, but we have the Hill function, which has now the k in the numerator instead of the activator that we had before. And here, instead of an A that we had before, we have an R, which is the representative of the repressor. But basically, is the same than we had before. And I can show you again the, sorry about this. The same app, but with the repressor. It's not this one, it's this one. As we see, the shape of the function is similar to the one before, but when there is no zero amount of molecules of the repressor, we have the maximum level of expression, which in the normalized case is 1. And as we increase the number of molecules of the repressor, depending on the value of this constant, here the constant KD, the half concentration constant, is to 250 more or less. If we increase the number up to that value, we have half of the repression. If we increase more the number of molecules of the repressor, we can reach a higher level of repression. How much repression can we get? That depends on the leakiness of the promoter, which is the value of alpha here, alpha 0. If we have 0 leakiness, it means that the promoter is an ideal promoter, that there is complete repression when we have enough amount of the repressor, we get to 0 amount of expression here. If we increase the value of the leakiness, a normal promoter in the lab will have between 20%, this number is 0.2, 0.1, 0.05. If it's a very bad promoter, it can have a higher number here, which it means that even if you have a lot of repressors, there still will be an amount of expression there. And again, if you increase the Hill coefficient, the steepness of the transition will be higher than before. Okay, so later you will be able to play with this and to get familiarized more with Hill functions and models. So to sum up, depending on if the promoter is an activator, an activated promoter or a repressed promoter, the action of the transcription factor will make expression go up as the number of activator transcription factor molecules go up. Or if it's a repressor, increasing the amount of the repressor transcription factor, it will lead to a reduction in the level of expression. The leakiness is related to this value here, the 0.2. Let me put the pointer. In this example, for example, this promoter has close to 0.2 of leakiness, meaning that even with the higher level of the transcription factor that represses the promoter, you will still have a residual amount of expression, which is called the leakiness of the promoter. Okay? Okay, let's go on to make the models easier. So what happens if this process that we had here before, the transcription, the conversion of between DNA and mRNA is a fast process. What is fast? It means that, let me remind you the equation, the ordinary differential equation that models the change in the amount of mRNA, which is this m dot. This is the derivative of m with respect of time. And if this is very fast, it means that we can set m dot, which is the derivative of the mRNA, to 0, because it's fast and it reaches the equilibrium really fast. So this means that if you remember from math, from differential equations, to obtain the equilibrium of this equation, which means the value that it will have when enough time happens and the changes reach a steady state value, this will be, we will get it doing m dot equal to 0, which is this guy, as I'm writing here, equal to 0. From there, in this equation, we can solve for the value of m. We can put it in the other side as a sum, and then divide by d1, and we will get an expression for the amount of mRNA that we will have in the equilibrium when the system reaches the equilibrium. But as we say that the system is fast, we know that this process is fast, we can calculate that now. And using that, we can go to the, so this is what I was saying before. If this reaction is fast, we can solve for the value of m, and we will get this expression. And if we go back to the equation of the protein, the differential equation that tells us how the protein changes in time, and instead of m, we plug this guy, which is the value that that m will have, which is here, we will get only one equation for our system, which is modeling how the amount of protein changes in time depending on, in this case, A. The only thing that changes here is A, which is the amount of activator that we put. This, for example, can be a molecule that we put from outside, or it can be another protein that is expressed in our circuit. All the other elements here, K2, D1, B0, etc., are fixed values, which are the parameters of this equation that depends on the construction of the circuit, but they don't change in time. The only one that is changing in time here is the A, which, as I said before, is the amount of activator. So if we solve this equation in MATLAB, for example, using ODE45, or there are many ways of solving a differential equation, we can do it by hand, but if the equation is more difficult, we can use a software to solve it. We will get this, for example, a plot which is like this one that I have down here, which I plot for different values of the activator. If I have a low level of activator, I will reach a low amount of molecules of the protein. If I increase the value of the activator, I will have the change in the proteins will go to a higher level. This, if we see the final value here, depending on the amount of activator that I added to the system, or that is in the system, we will have different levels of the amount of protein that we have in our circuit. The leaky promoter will have some expression in the absence of induction. If the leaky is used for a repressor, so that means that if you put a lot of transcription factor repressing that promoter, you can still have expression. When we talk about activators, it's called basal expression, and is what you are writing there, Martin. And it's the expression that you have when there is no activator in the system. Okay. Okay. So what can we do with this? Let's relate it a little bit with the work that we will do in the lab. If we have an experiment, we can put a fixed amount of activator in our system and start to measure some way. We will see different ways of measuring later, and the measurement committee is devoted to help you in measuring parts of your systems or everything that we can do. So if we start the experiment, for example, we hit the system with the fixed amount of activator, and a number of molecules of this protein start to increase until it reaches a steady state value, which is here in this axis we have time, different than the ones that we had before. So what we can do is, with a plate reader, for example, we can measure the amount of protein. If it's a fluorescent protein, we can measure it in time, and then we can come to the model and compare it with the solution of these equations that we have here. There, we will have a way of putting together the experiments and the results coming from the model. We are also planning on doing a webinar on this later in the summer. So if you like that part, then keep tuned. So, sorry, if we look at this value, the value that we get when we reach the equilibrium, means when the value of the protein stops moving. Sorry. For different number of amounts of the activator concentration, we can do different experiments with different amounts, increasing the amount of activator, and we can measure the number of molecules that we have in all different experiments. And then we can go and do the same with the model. We can set different values for the activator, for AN, and plot the result, and then we can combine them together. And another way, like we were talking before, this is another way that we can use to combine experimental and data and what we got from the model. So in this way, having the equation of the protein, mathematically, so in the experiment, what we do is we wait enough time until the concentration of the protein reaches the equilibrium. Mathematically, what we do is we set the derivative of the protein, which is P dot here, to 0, and we solve for the value of P. So when we solve, what we have here on this side is very similar to the Hill function that I was showing you before. That was the importance of showing you that at that point, because here all these values, K2, D2, D1, these are fixed and depend on the circuit that you have. And the only one that will change is that you can change is the amount of the activator, which it will give you this this shape of the Hill function. Okay. So in this way, we can relate the output of our model, which in this case can be the red curve, which is representing the amount of protein for different values of the activator. We can relate it with the result that we can get in the lab doing the same kind of experiment. Okay, so a very short, I'm getting close to the time. These are 3 slides that I will show very, very quickly because I would like to go to the rest of the presentation. We are getting short with time. For example, imagine that you have a circuit like this that many of you may know is using the quorum sensing system, AHL, which goes in and out the cell and binds the transcription factor lux R, which when you have two of them, they dimerize, they repress this promoter, which make the amount of this protein go down. And here we have two proteins. One is a protein of interest that can be GFP, and the second one we have here is LAX-I, which in turn is the synthase of AHL. So what it means that if you have higher level of this, you will have higher level of this. It will produce more AHL, and this will repress this promoter, which will make the amount of LAX-I and GFP go down. This is implementing a negative feedback loop around LAX-I and AHL. So, but basically what I want to tell you is from this, you can write all your reactions, which in this case, we have 22 reactions and 10 species. And we can go there and make our model, which will end in this case with 9 or 10, sorry, differential equations. And finding values for these parameters, we can simulate how our system will go. And if we can see if it works, if it doesn't work, or what values of the parameters we will need to have in order to have the function that we wanted to have. So it's just an example. But the interesting part of the example is that from this bigger model that we have here with 9 equations, we can use some assumptions like the ones we used before, for example, that the RNA is fast, and we can get rid of some of the equations and obtain a smaller model that will be more easy to handle and to simulate. And here, just for showing you, a comparison between those two models for some set of parameters. The red line is the reduced model, and the dotted blue line is the complete one. As you can see, there are some difference in the dynamics, in the changes, but then when they reach the steady state, all the two models, the complete and the reduced, go to the same value. So the bottom line here, what I want to tell you is that it's okay to drop some complexity and to simplify our models, to have models that are more easy to handle, but we just need to know what we are doing, okay? So this one is more, now we change to a part which is more related directly to the measurement committee, and is we imagine that we have our model, we have values for the different proteins and concentrations, and we want to relate that to the lab. So how can we measure what happens in the lab? And fluorescence, of course, is a good way to measure what happens inside the cell. It was discovered a few years back now, but the original fluorescent protein, it was the green fluorescent protein from the jellyfish Aquaria Victoria. And after this one, which was the first one, sorry. So this one, as you can see here, is a microscope image of some, I think it was HeLa cells, and here is some protein purified. You can see it, so we agree that it's a good reporter. But there are many other fluorescent proteins, and if you were in the previous webinars of the measurement committee, we mentioned already that we have a resource here in the measurement page, in the measurement hub, which can help you to select the right fluorescent protein for your project. There are many different fluorescent proteins, and when you have to select one or more than one, you need to study the different the emission and excitation spectra of the proteins and see which one is better for your project. And what happens if we calibrate the measurement, as Jake was mentioning this morning? If we make a calibration, so we can know what the value coming from the plate reader means in terms of numbers of molecules of some fluorescent reporter standard. In the case of the green proteins, we use fluorescein, you have all the information in the page here. And this year we are also including a standard for red proteins, the Texas red. If you calibrate your fluorescence measurement with these tools, and also if you calibrate the optical density, the OD, with the beds, with the particles, that tells you, that relate the number of OD that you measure with an amount of cells that you have in your culture, you can have an even better way to know what happened in the cell. And how do we relate that with the model? That's the interesting part. If you do this calibration that I was talking before, this, if you look at this one, it's a growth curve of a culture that I have here calibrated in number of particles. Here I have a fluorescent measurement from a plate reader, also calibrated with fluorescein in numbers of molecules of equivalent fluorescein. If we put these together, we can get a unit which is the number of molecules of equivalent fluorescent per particle. And this is a unit that is equivalent to, the number of molecules that we have inside the cell. And that is what we were modeling with our P. Remember in the previous part of the presentation, P was the number of molecules of the protein that we have inside one cell. So here is the way that we will have to relate the experimental data that we have, if you have it calibrated, with the output that we get from the model. So with these two together, we can effectively go back from here in the cycle. Calibration and measurement will help us to go from measuring our system back into the design and modeling and simulation, where we can feed back the information that we got from our constructed circuit. Okay. And with that, we reach the end of the webinar. Thank you for listening. This was the last one from the measurement committee. As we said before, and Alexis wrote there in the chat, the measurement committee will be actively working to prepare different workshops this summer, so stay tuned. Fill the survey that we had here, and also the other survey that we will post, I think, in the measurement committee web, in the measurement hub, about which kind of webinar you will like to be hearing this summer. And that's it. Thank you for everything. So let me check a few of the questions that we have here, so we can try to answer them, or we can get in contact so we can answer them effectively later. I see some questions related to artificial intelligence. Where was it? I don't see where is it now. But okay, what I can tell you about artificial intelligence is that, so now artificial intelligence involves many things, but one of the things that is very, very useful these days is deep learning or machine learning, which is basically neural networks that you can train with data to obtain a prediction of the model. So that's a different approach of modeling. It's also useful, but that one involves having much more data. That's the reason why nowadays they are very trendy, is because there is the amount of data in general that we have is much bigger than before. But still, the amount of data you need, it's very big. On the other hand, our models, so those models rely on data. The models that we are doing here are based on first principles. They have different coarse grain, but depending on the level of interest that you have, you can explain mechanistically the different aspects of what happens inside the cells. So what we are trying to do here is more related to the first principles and the mechanics that are behind. If you use deep learning or something like that, that's what is called the black box model. You put the data and you get the prediction, but it doesn't explain you what the system is doing in terms of the function. Okay, I hope I answered that question. I saw a question about recommending literature. We will write a section in the page where we recommend the literature. There are many, to my mind. There is one which is called Mathematical Biology from Murray. Okay, but we will write a section about different literature that you can reach related to this. What else? Definitively, it's very difficult to reach the questions here in the chat because there are many. But definitively, you can reach us via the Measurement Hub, via email, via Slack, and you can ask the questions again, and we will try to answer you. Okay. Thank you, and thank you, Roderick, for hosting. We will hope that you have an exceptional iGEM year, and keep in touch and keep tuned to the measurement committee. Thank you.